myself, this is Adhav from Prof College, Srinagar. Today we will discuss about respiratory system of T.Y.B.Sc. T.Y.B.Sc, due to amphibious mode of respiration, it shows dual habitat, and hence there are two respiratory organs. For water, that is aquatic respiration, there is the presence of single gill, or it is also called as ctenidium. And for land respiration or for terrestrial respiration, the animal shows presence of pulmonary sac or lung. Thus, these are the respiratory organs. First, ctenidium or gill for aquatic respiration. And pulmonary sac or lung for land or terrestrial respiration. Then, besides this, there are also right and left nuchal lobes. This is the left nuchal lobe, this is the right nuchal lobe, this is the mouth surrounded by pair of tentacles. First pair of tentacles, second pair of tentacles, these are the eyes, this is the head. When there is need of respiration at that time, head and foot protrudes out from the shell, and the animal shows respiration with the help of these nuchal lobes. Nuchal lobes are nothing but the fleshing or contractile processes of the mantle itself. On either side of this head, these two processes called as right nuchal lobe, which is short. It is also called as pseudopodium or right siphon or right nuchal lobe. Then on the left side of the body, there is second fleshy lobe. It is called as the left nuchal lobe or left siphon or left pseudopodium, which is comparatively larger compared to this right nuchal lobe. These are the respiratory organs. First, we will see the ctenidium or gill, which is the organ for aquatic respiration. This is the diagram of ctenidium or gill. Ctenidium or gill attached to the body wall on the left side. Left side here. This ctenidium attached to the left side. Ctenidium or gill is the organ of aquatic respiration, and it is present in the right side. This right side wall of mantle having the attachment. That attachment of gill is called as line of attachment or ctenidial axis. It is also called as line of attachment. Means, this is the mantle cavity. Suppose this is the mantle cavity of the body. This mantle cavity of the body is divided into two unequal lobes or two unequal chambers due to the presence of epithelium. This is the epithelium. This epithelium we have seen in first external characters. This epithelium is nothing but the skin fold. This skin fold, which arises from right nuchal lobe and goes backward up to the end of mantle cavity. Due to the presence of this epithelium, this mantle cavity divided into two parts. Right small chamber is called as branchial chamber, into which present the gill or ctenidium. This is the location of gill or ctenidium attached to the mantle wall by means of this line of attachment. It is called as ctenidial axis. And upper or opposite to this epithelium, large chamber, large left side chamber is called as pulmonary chamber, into which pulmonary sac is present. This pulmonary sac present on the roof of mantle cavity. We will see first the ctenidium. in detail. Ctenidium or gill attached to the right side mantle wall by means of this ctenidial axis. This ctenidium shows only one side gill lamellae, hence it is also called as monopectinate gill. Monopectinate gill. If both the side lamellae, then we call it as a bipectinate type of gill. But here in phylum, only single side gill lamellae attached. Hence, this type of gill or ctenidium is called as monopectinate type of gill. These gill lamellae are largest in the middle, but they gradually become smaller towards both the ends. These lamellae are having numerous small hair-like processes. These small hair-like processes are called as cilia. These cilia are responsible for continuous beating of water current in the surrounding medium, and hence water enters through left buccal lobe and goes out through right buccal lobe. Single ctenidium looks like this. Here there are four leaflets or four lamellae. This is the single lamellae. This is second, third, and fourth. Here there are four lamellae. You can see in this diagram. Each lamellae having broad end attached to this ctenidial axis, and narrow end is free. It is free in the branchial chamber. Means this is the narrow end, this is the broad end. This broad end attached to the ctenidial axis and narrow end is free. These free ends are here in the branchial cavity. They are free. This ctenidium having afferent side and efferent side. The largest side of each lamellae is called as efferent side, and small side called as the afferent side, which is present on the right side. This is present on the left side. Each lamellae or each leaflet, we may call it as a leaflet or lamellae. Each lamellae on its both the surfaces. Suppose this is single lamella. This anterior surface and posterior surface, both the surfaces are provided with transverse ridge or grooves. These transverse ridge or grooves are called as pleats. These transverse grooves or pleats become smaller towards the apex and they are larger towards the base. These transverse pleats or ridges or grooves, they are richly supplied with blood vessels which contains the oxygenated type of blood. Then each lamella shows beating due to the presence of this cilia. Here you can see when water enters through left buccal lobe. When water enters through this left buccal lobe, here there is the leaf-like process called as the ostradium. This ostradium serves to test the nature of water. This water enters into this branchial chamber. At that time, this epithelium closes this flap, that is the pulmonary chamber is closed. Water flows towards this branchial chamber where gill or ctenidium is present. All the lamellae bathed in this water current, and here exchange of O2 and CO2 takes place between blood vessels which contains deoxygenated blood, and this water, incoming water, contains fresh O2 or dissolved O2. Here there is the exchange of O2 and CO2 takes place because each lamellae having thin vascular and permeable mucous membrane, due to which there is the exchange of O2 and CO2 takes place. Means blood vessels which are richly supplied to these plates, transverse plates, they contains deoxygenated water, deoxygenated blood, means CO2 is present here, and this incoming water contains dissolved O2, dissolved oxygen. When water current enters through left buccal lobe, all these lamellae bathed and each lamellae having these transverse plates, all they contains deoxygenated blood. Here O2 is absorbed due to the thin membrane and CO2 is driven out, or there is the exchange of O2 and CO2, and this CO2 goes outside through outgoing water current through this right buccal lobe or right pseudopodium or right siphon. Means water enters through left buccal lobe. Here there is the ospiradium which tests the nature of water. Then this water flow enters into this branchial chamber where gill or cnidium is present, and each lamellae or each gill lamellae or each lamellae or leaflets provided with these transverse plates which having deoxygenated blood vessels, and there is the exchange of O2 and CO2. This type of respiration is called as aquatic respiration. When animal is inside water, at that time this type of respiration is performed by the phylum Globosa. Then second, the pulmonary sac or lung. We will see here. Pulmonary sac or lung is present in the pulmonary chamber. It is present in the pulmonary chamber. This pulmonary sac is nothing but the modification of mantle itself. Here there is a bag-like structure which is present at the roof of mantle cavity. This bag-like structure or which is present on the roof of the mantle, this bag-like structure opens into pulmonary chamber by means of an aperture. This aperture is called as pneumostome. It is called as pneumostome. Means this pulmonary sac which opens into pulmonary chamber by means of a small aperture. This aperture is called as pneumostome. This pneumostome is guarded by unequal valves. The ventral wall of pulmonary sac is creamy white in color, and dorsal wall of pulmonary sac is highly pigmented. This pulmonary sac internally provided with network of blood vessels which contains deoxygenated blood, means CO2 content blood. When animal lives on land or when it glides on land, or when sometimes if water is dirty or deficient water is present, at that time the animal projects its left siphon above the water level, about 2 cm it can be projected, or sometimes when it lives on the land they can respire from the atmosphere. The air is drawn through this left buccal lobe. Air comes through this pneumostome or aperture of pulmonary sac. Air drawn into this pulmonary sac. The wall of pulmonary sac shows contraction and relaxation. That is, it is highly contractile. Here, when air comes through this pneumostome, the blood vessels which are present here, they contain CO2. Here, the wall of pulmonary sac is again thin, vascular, highly contractile, due to which O2 from atmosphere enters through this wall. blood becomes pure and CO2 from that blood goes out or driven outside again from the left nuchal lobe. Means there is the exchange of O2 and CO2 takes place within blood vessels and fresh O2 from the air. This is the pulmonary sac or lung, which helps for land respiration or which helps for terrestrial respiration, or sometimes when aquatic vegetation is present in the surrounding water, and if water is having less O2 or dirty water is present in which the animal lives, at that time they can protrude their left nuchal lobe outside the water, and animal is submerged inside that water. At that time also they can respire with the help of this type of pulmonary sac or lung for atmospheric air, and exchange of O2 and CO2 takes place there. Then right and left nuchal lobes also play important role. How? Here the epithenia, they press, this epithenia is pressed to close this chamber or to entry of water to left nuchal lobe. When it enters through this way, left nuchal lobe, it again goes towards the right side, that is the bronchial chamber, and there is the exchange of O2 and CO2. Means the epithenia plays important role for both the respiration.